I want you to take a moment and imagine that you're blind. How would you have gotten here today? Could you have eaten breakfast on your own? Could you have even found this room? Vision is the primary way that we interact with the world, yet every year hundreds of thousands of people are stricken with diseases like macular degeneration and retinitis pigmentosa, leaving them completely blind, drastically lowering their quality of life. Now, vision is a fickle process. There are a lot of little things that have to go right for you to actually be able to perceive the world around you. And the main place where things tend to go wrong in disease is at the interaction between your photoreceptors, so your rods and cones, and a cell type called the RPE. At the back of your eye, in your retina, you have a layer of these rods and cones, and these are the cells responsible for taking in light information and sending it to your brain. So when you lose these cells, you lose your vision. Directly behind those, you have a layer of these RPE cells, and the job of the RPE is to keep your photoreceptors happy and healthy. And they actually do that by physically ingesting pieces of your rods and cones, breaking down their contents, and recycling them for future use. So we've actually known about this process since the 80s, but even still, we don't have a clear picture of even how it works, let alone why it might go awry in disease. So my research really aims at utilizing new technology to better understand this process. So what we can actually do is take 3D movies of this ingestion taking place. So in this movie I'm about to show you, the green is the RPE cell, and the red is a piece of rod photoreceptor. And as you can see, the RPE makes this physical cup and engulfs the piece of rod, pulling it in for degradation and recycling. So I have some preliminary data that shows that by modifying outside factors, things like temperature or things like the density of these RPE cells, we can totally change the efficiency of this process and even change the physical way that this cup forms. I've also been utilizing a number of drug treatments that others have been using in similar systems as a way of really picking apart and modifying the different proteins involved with this process to really try and get a better understanding of how this ingestion works. And I want you guys all to remember that this process, it's perfectly normal. It happens in everyday life, but for some reason, when this process breaks down, your rods and cones start to die and you lose your vision. And that's what's actually happening in a lot of these major diseases that cause blindness. I really hope that by taking these 3D movies and by modifying these outside factors and using these drug treatments, I can really get a clear picture of exactly how this process takes place. And more importantly, try and figure out clues as to how and why this process might go awry in disease. I really hope that by doing this, I can find new insights into vision science and find ways to prevent blindness in the future.